The overall goal of this experiment is to observe the endo and exocytosis cycling of synaptic vesicles on the live preparation of Drosophila larval neuromuscular junction using fluorescent FM dyes.
This method can help answer key questions in the field of neuroscience, such as which genes are critical for the function of the synaptic vesicle cycle. The main advantage of this technique is that you can optically track vesicle cycling in a live preparation using 3 different types of stimuli: high potassium depolarization, electrical stimulation, or using optogenetics.
To begin this procedure, add the anti-HRP antibody conjugated to Alexaflor 647 to the dissecting saline. Next, add an elastomer-coated cover slip to the dissection chamber, and then add the dissection solution with HRP antibody for labeling the NMJ presynaptic terminal during dissection. Using a fine paintbrush, transfer a wandering third instar from the food vial onto an elastomer-coated cover glass. Then, fill a glass micropipette tip with a small volume of glue using negative air pressure generated by mouth. Subsequently, position the larva dorsal side up with forceps and glue the head to the elastomer-coated cover slip with a small drop of glue using positive air pressure by mouth. Repeat this procedure with the posterior end of the larva, making sure that the animal is stretched taut between the 2 glue attachments. Using scissors, make a 1 mm horizontal cut at posterior and a vertical cut along the dorsal midline. Then gently remove the dorsal trachea, gut, fat body, and other internal organs covering the musculature. Repeat the gluing procedure for the